Hi everyone! Today we're going over eukaryotic cell organelles. You are going to be doing a project with this later, so make sure you take good detailed notes and let's get started. Endoplasmic reticulum. This is also called the ER, and we do have two types of it. The smooth endoplasmic reticulum does not have any ribosomes and is in charge of making lipids. So lipids being the fats, we saw the triglycerides, the phospholipids, steroids, hormones, all of those things. So the smooth ER mainly makes those. It does contain lots of enzymes which help that out, but a bulk of your proteins are created at the rough endoplasmic reticulum. So that's the RER. The rough endoplasmic reticulum gets its name because it is studded and it has tons of ribosomes that are on it, and the ribosomes are the little guys that crank out the proteins during translation. So ribosomes, just talking about this, we find them on the endoplasmic reticulum, and they also just kind of like float around within the cell cytoplasm. So we have the attached ones to the rough ER, we have the free ones floating around in the cytoplasm, and they contain a couple of different parts that we're going to look at here in class. The first one is just like regular protein components, and then we have an RNA molecule. So that is the rRNA. We are going to be talking about this later on. Now the rRNA there, the analogy that we're going to be using later on is that this is like the structure. It gives like the baseboards for everything, which is going to end up helping other RNA molecules pair with it and then help align the amino acids in the proper sequence. The Golgi apparatus. This is tons of stacks of like membrane-bound sacs, so I like to think of it looking like tons of little pancakes stacked on top of each other. It takes in the proteins either from the free-floating ribosomes or from the rough ER, and then it packages and then delivers the final proteins. So in class we talked about how polypeptide chains are the polymers for proteins, but the proteins go from like polypeptide to protein through tons of folding. There's 4 steps. This is where a lot of that is going to happen in order to refine and get that final protein put together. So everything arrives to the Golgi enclosed in these things called vesicles, which is when we have another like phospholipid bilayer. And those vesicles, they attach with the Golgi apparatus. The Golgi apparatus then is able to package it, finally like take care of like the end details, and then the proteins leave out of the Golgi apparatus after being chemically modified and then get shipped off to wherever they need to go. So I like to think about the Golgi apparatus being like Amazon Prime. I can order tons of things through there. Amazon is the main headquarters that takes care of everything. They probably like refine and like take care of the last few details with some products, and then they're in charge of shipping them all out and getting them to the proper places. So this is a solid example of how we see proteins being produced and then sent out of the cell. Over there in the purple, we start out with the nucleus, and the nucleus has its own nuclear envelope. So this is what encloses the nucleus. And right outside of it, this is going to be our rough ER, and you can tell it's rough because it's got the ribosomes. So I think about the ribosomes and the rough, the 2 R's helps me keep it straight. And then over here to the side, it's the same endoplasmic reticulum, but it just does not have ribosomes. So that is the smooth endoplasmic reticulum. So these proteins are created. They ended up getting put into like a little vesicle, a little like packaging unit. And then that gets transferred all the way over to the Golgi apparatus. And then the Golgi apparatus is going to like fuse with that vesicle. It's going to modify and take care of the proteins, make sure that they are in the correct conformation, and then send them all out. And this is an example of a protein that gets expelled from the cell. So the one thing you remember from biology, the mighty mitochondria. We're just talking about this so much more next week. But this is the site of cellular respiration. You guys are going to have to know this thing front and back. Cool things about the mitochondria, they can reproduce on their own and they contain their own DNA. And because these are found within the cytoplasm, all of your mitochondria come from your mother. The only thing you get from your father is that original DNA. So you get everything that is outside of the nucleus, everything that is within the cytoplasm, including the mitochondria, from your mom. So they have their own DNA, and so there's something called like a, oh, I forget what it's called. Doesn't matter. Maternal DNA thing, where we can find like females that we all have common ancestor with just by tracing mitochondria. It does have an inner and an outer membrane. This is another piece of evidence that supports that the mitochondria was some form of like bacteria that then created a symbiotic relationship with a eukaryotic cell. The inner space in between, it's in that nice like light blue there. That's called the matrix. And then many mitochondria, we find so many of these in muscle cells. Now, if you are like an endurance runner, chances are you're going to have more mitochondria in your muscles than I will. If you are a weightlifter, you're going to have a different type of muscle density, and you may have less mitochondria. So really, the more aerobic exercise you do, the more long distance, endurance things, the more mitochondria you're going to have. So then lysosomes and peroxisomes. Lysosomes, they are, once again, phospholipid bilayer. We've got that plasma membrane. And these are the things that end up like breaking down nutrient molecules that we don't need anymore and getting rid of like excess particles and then broken organelles, things that just aren't efficient anymore. And then our peroxisomes, these are in charge of like creating bile, detoxing a lot of things, including alcohol. Which means that they are abundant in the liver and in the kidney. Oh, that picture got covered up. Okay. So yeah, tons of them in the liver. They're going to help clean out and get rid of just like tons of stuff that should not be there. Cilia and flagella, now we're talking about movement. So cilia, we're going to see a majority of the cilia in this class that we talk about in your respiratory tract. You have cilia in the respiratory tract. They line the airways. And they move back and forth just like little hairs, and they sweep dust particles up and out of the respiratory system so it doesn't get caught in the lungs. If you're a smoker, then that smoking ends up like kind of putting the cilia to sleep, which means that you are susceptible to dust and really just anything getting into your lungs, which is why people often experience something called smoker's cough, which is when the cilia like wake up and then they look around and they're like, oh God, there's so much stuff here, and then trying to sweep it all out. So cilia, it aids in movement, it aids in moving fluids, and for lots of bacteria, it's just how they get around. Same thing for flagella. It's a tail-like extension. We've got like some nice little eukaryotic organisms down there, but they help propel the motion. They are the tail that moves it in one way. Humans have flagella in the sperm cells. It's on the sperm cell. So that is a stereotypical example, and as a result, like this is how sperm get from A to B. There are tons of mitochondria packed around like the base of the flagella so that the sperm cell can propel itself forward, but nothing within the female reproductive system or system at all that I can think of that has flagella. Although our fallopian tubes are lined with cilia to help the eggs float down. Vacuoles, vesicles, these are just those membrane-bound sacs formed by the cell membrane. So once again, a phospholipid bilayer. The vesicles, vacuoles, this is what we found going from like the endoplasmic reticulum to the Golgi apparatus with those proteins. And they are used to store just about anything under the sun. Macromolecules, water, sometimes toxins if something is poisonous, and other organisms, and lots of pigments get stored there as well. So microtubules and filaments, these are important for the cytoskeleton. Cyto means cells, skeleton is referring to skeleton. Microfilaments, it's tons of like actin protein. That'll be relevant when we talk about muscles. And it allows for the cells to move, contract, and really just like hold themselves in place. More proteins, just a little thicker. So our nucleus, oh my gosh, so much. It houses all of the genetic material for us. All of our DNA is found within there. It directs all cells' activities. It is the main hub. It controls everything. It is enclosed by its own phospholipid bilayer. And it has like these little itty bitty pores which allow for like the RNA to escape as well as some other proteins. Within the nucleus, we have a smaller, denser region, which we call the nucleolus. And this is where it's a dense organelle made of RNA that it gets covered up, made of RNA and protein, and this ends up forming the ribosomes that we talked about earlier. And then we also have chromatin there, which is a spaghetti form of DNA. And all of that stuff just kind of hangs out in that one central hub, staying nice and safe. And that is it. Make sure that you have all the directions and that you're able to work in your group with the project for the rest of the day. And if you have any questions, I am in the building, so feel free to message me on Schoology. Have a good day.